Hi, my name is Hayden Blan, and my presentation is called Cyanobacterial Toxins in Freshwater Environments. And it's a review on the toxin microcystin and its toxicological properties. So first of all, what is cyanobacteria? They're commonly referred to as blue-green algae. They make up a group of prokaryotic photoautotrophic bacteria, and they can populate in several terrestrial and aquatic environments, and they have large morphological diversity. They can produce thick mats that impede recreational activities, swimming, fishing, and they can cause odor problems and oxygen depletion. They can often be confused with green algae, which are not thought to produce toxins. However, the cyanotoxins can form harmful blooms and produce potent toxins called cyanotoxins. They are found naturally in the environment and do play an important role in nutrient cycling, but problems can quickly arise during a process called eutrophication. And eutrophication is naturally a slow process, but it's exacerbated when water bodies receive excess nutrients that stimulate plant growth like algae, periphyton, and noxious weed plants. Increasing anthropogenic activity has caused more severe and frequent cyanobacterial blooms, and consuming tissues or supplements uh, that have accumulated toxins, drinking contaminated water, recreational activities are the main points of exposure and can pose a health risk. So microcystins were found to be the most reported cyanotoxin in fresh water by an extensive review of cyanobacterial blooms worldwide. They degrade water clarity and oxygen content and has detrimental effects on aquatic habitats. They also have the capability of producing harmful secondary metabolites called cyanotoxins, which are grouped as hepatotoxins, neurotoxins, and dermatoxins. The most common and possibly the most toxic variant found in the environment is Microcystin LR, or Microcystin leucine arginine. It has an abundance of toxicological data. It's the only variant with human exposure guidance information. Uh, they can potentially lead to acute or chronic health issues via increasing reactive oxygen species and the inhibition of serine threonine protein phosphatases. The liver is a typical target of Microcystin LR, although it has demonstrated neurotoxicity in several other studies. They're most commonly found in freshwater lakes and ponds, and they make up a group of 80 cyclic hepatopeptide toxins, and they're produced by the bacteria from which it was first isolated. Oh, yes, Microcystis aeruginosa. My apologies. The purpose of this presentation is to examine the toxin microcystin and highlight its sources, pathways, receptors, controls, in freshwater environments. So microcystins can be produced by several species of cyanobacteria, um, Anabina, Fissurella, Nodularia, Oscillotera, and several members of Microcystis are capable of producing it. They live in the water column and compete with other single-celled algal species for niche space. Many forms of this toxic uh, species have adaptations leading to their proliferation in bloom event. Uh, Although oral route is the main source of exposure, there can be uh, dermal and respiratory routes. They're usually non-threatening and have effects like contact dermatitis. They can also bioaccumulate in common invertebrates and vertebrates, zooplankton, mussels, and fish, and that can be passed up to higher trophic levels like animals and humans. Next is this is a picture from the EPA's website, and its key factors related to the proliferation of harmful algal blooms, frequency, and toxicity. So, urban and agricultural runoff can be significant sources of nutrient inputs. Warm surface water and optimal light can increase blooms as well. And, uh, the toxins can be transported downstream, and water filtration for uh, drinking water intake facilities don't always filter out said toxins in normal processes. Uh, Microcystin is usually bound within the bacteria itself, within its cell structure, and problems begin to arise during the collapse of the bloom event, where the cells die and it's released into the surroundings. Microcystin has multiple pathways that elicit toxic responses. They're large hydrophilic molecules believed to enter liver cells via the basolateral organic anion transporter peptides, or ATPs. Once they're transported, toxicity is exerted by inhibition of serine, threonine protein phosphatases 1 and 2A. And this can lead to excess phosphorylation of structural filaments, degrade the cytoskeleton, and break down the hepatic ultrastructure. Current research has pointed to genotoxic properties, such things like the nucleotide excision repair and DNA double-strand break repair by non-homologous and end joining. They have been shown to be inhibited by microcystin. Calcium modulin-dependent multifunction protein kinase 2 and the nuclear phosphoprotein p53 can both be activated, and there are potential routes for apoptosis through the involvement of outer membrane permeabilization and transition of the mitochondria. Production of reactive oxygen species is a biochemical feature of microcystin toxicity. It's been observed in several in vivo studies in mice and rat liver, heart, and reproductive systems, and in vitro systems like human hepatoma cell line HepG2, fish cell lines RTG2, and PLHC1, lymphocytes, and human erythrocytes. Oxidative stress is related to mitochondrial metabolism, and the surge in calcium disrupts the membrane, causing cell death and reactive oxygen species. They can also cause cytoskeleton disruption and change in cell morphology by reorganizing the microfilaments, intermediate filaments, and microtubules, leading to intercellular interactions and deterioration in cell morphology. This is showing multiple in vitro and in vivo experiments indicating multiple in vitro and in vivo experiments indicate low intestinal absorption for microcystins, and oral consumption is the most common route of microcystin exposure. Microcystin LR is water soluble and is considered an environmental health concern, and human exposure is recognized as a worldwide health issue. They are large cyclic peptides that require transporters to diffuse, and once ingested, most of it's absorbed into the ileum, to the lesser extent the jejunum of the small intestines, where the OATPs can receive the microcystin and are transported to the liver. It also has port efflux by multi-drug resistant protein 2. It explains the accumulation and the distribution in the liver. And this flow chart was showing this oral route, and showing some elimination from the liver and kidney, but a very small percentage being at 15 and 10% respectively, um, which is why it can be introduced to the systemic circulation and be introduced to the kidney, brain, lungs, heart, reproductive organs, and can cross the blood-brain barrier. And that flow chart shows that absorption, distribution, metabolism, and excretion after oral exposure. The occurrence of cyanobacteria and their associated toxins in waters to which people and animals may be exposed depends largely on the conditions of their respective water body and the extent at which they favor the proliferation of said bacteria. In natural water, dilution is the major process by which toxicity is reduced. After a bloom event or when the cells of microcystin ruptured, their toxins are released to the environment. It can be diluted, but often are not reduced below the critical point for organisms. It's usually in an isolated area where that animal or person would be in contact with. Phosphorus and nitrogen are the main macronutrients considered when deciding limiting factors for biomass, and in turn cyanobacteria in each water body. Naturally, microcystins can be absorbed onto sediments and can be broken down by photodegradation. Drinking water treatment is the last line of defense in delivering safe, clean water to consumers, but it's not necessarily good control for exposure in the environment. The proactive risk assessment and management framework can be applied to identify and manage public health risks from water use systems, including microsystems and water safety planning. Regular monitoring is the most effective in monitoring these blooms and is focused on water bodies in which these bacteria are likely to occur, but it is challenging to detect. There are different treatment options. Degradation were shown in a week in a laboratory setting over 12 days in sterilized reservoir water and over 27 days in deionized water. It was found that UV light exposure rapidly degrades microcystin LR. Physical processes, including membrane filtration and absorption by activated carbon, can be used. The chemical processes using chlorine, ozone, and other oxidants give a better solution. Some biological methods have been used, like using the crucified Ochromonas danica and the microbes and other microbes, but are in limited use due to long residence times. Biofiltration with naturally occurring bacteria was shown to take over 7 days for any degradation to appear. There was also a report that showed potential uses of barley straw for cyanobacterial control, especially M. aeruginosa, by significantly inhibiting their growth rate due to their phenolic content. So further discussion, the preceding literature identified some of the major sources, pathways, receptors, and controls of the toxin microcystin in the environment, and highlighted many areas of its toxicological effects. It is produced by cyanobacteria that can be found in almost every habitat around the world. Although this bacterium is harmful, it's a natural part of the environment that takes part in nutrient cycling and even contributed to the formation of Earth's aerobic environment. They can be produced by several species and typically only cause problems during bloom events upon cell death or disruption of the membranes. Current levels rarely exceed drinking water guidelines, but the combination of multiple sources, along with increasing global temperatures and climate change, could exacerbate this problem in the future. The liver is the main target organ of toxicity, followed by the kidney. Several organs can be negatively affected. Mentioned above, acute toxicity and death is rare, but it's currently thought that there are potential carcinogenic effects at very low levels of exposure or below the guidelines. The EPA determined there is inadequate evidence to assess carcinogenic potential of microcystins in humans due to the limitations in a few available human studies there are, which highlighted the need for more research. Currently, the best line of defense in combating this issue is proper management practices of water bodies. Regular monitoring is a key aspect, along with timely treatment and management practices that are safe for the environment, especially for potable drinking waters. They can easily proliferate in the environment, and proper uh, assessments, along with water safety plans being implemented effectively, will be important, not just day-to-day monitoring. There is currently a lack of clear understanding of human toxicokinetics of microcystins and the different aspects of absorption, distribution, metabolism, excretion, and future research is needed to understand the distribution in humans. And that was my presentation. Here are my references. Thank you. Have a good day.